In the life sciences in the Department of Molecular Biology at Princeton University. Her research interests include telomeres, DNA, helicases, I hope she tells us what all these things are, chromosome stability, and genome integrity. Professor Zakian received her PhD in biology from Yale, held several postdoctoral and research fellowship positions, and joined Princeton University in 1995 as a professor of molecular biology. Professor Zakian is a fellow in the American Academy for the Advancement of Science and is also a fellow in the American Academy of Microbiology. She has received many, many honors for her research and for her work to benefit women in science, including the Senior Women Award from the American Society of Cell Biology. She serves or has served as editor, associate editor, and a member of the editorial boards of prestigious refereed scientific journals such as Plasmid, Chromosoma, the Journal of Experimental Zoology, Trends in Cell Biology, Molecular and Cellular Biology, Genes to Cells, Molecular Biology of the Cell. And those are just a few of the journals she's been really good with. Professor Zakian has been very generous with her time with students, and as a professor myself, I know how much time and effort and reward working with students can bring. She's mentored over 100 undergraduate, doctoral, and postdoctoral students, which means that she has influenced the lives of thousands of students as her students go on to mentor other students. She has published over 150 scientific articles in scholarly journals and is active in presenting her research at scholarly conferences all over the world. And while doing all of this, she has served on many, many committees for Princeton University. And of course, she teaches courses in her area of specialization. She's given keynote addresses and lectures all over the world, and right here in Princeton as well. And I was quite touched, I must say, to see that on her lengthy and impressive curriculum vitae, she has also listed tonight's lecture, which puts the Princeton Adult School and you as her audience in very impressive company. Professor Zakian will speak to us about Joseph G. Gall, a scientist who helped change the definition of scientist. And I'd like to present Professor Zakian with an official 75th anniversary t-shirt. And if we got the size wrong, we have a few more to change them. You don't have to wear it for your lecture. Thank you so much. It's wonderful. We really appreciate your sharing your wisdom with us tonight. Thank you very much.
Okay, well, thank you all for coming out on this cold night. So I'm going to be talking to you today about Joseph G. Gall, Joe, who was my PhD supervisor from 1971 till 1975 in the Department of Biology at Yale University. I hope I'm going to be able to convince you that he's a really good topic for this great seminar series, which all the topics just sound so interesting. I can't believe I missed them. But, so why have I chosen Joe? I've chosen Joe in part, I'm sorry, but I'm probably going to have a little bit of vocal problem today with coughing and sneezing. What can I say? It's that time of year. No, thank you. I'll probably choke on it. I've chosen him in part because he's a great scientist. And I hope to give you a flavor for why I say that. But the major reason I've chosen him is because by example and by leadership, he has changed the composition of people who do science from a really almost totally male-dominated field to a field where there are now a lot of women in the life sciences. This is the 75th birthday of this organization. In April, Joe celebrated his 85th birthday. I was co-chair of a group that held a symposium in honor of his birthday, gathered speakers, former lab members, other scientists who've been influenced by him to talk about their work and about their experiences in Joe's lab. And Joe himself gave a talk on his work because he still works full-time every day. And as far as I can tell, the only thing different in the way he looks and acts compared to when I was in his lab in the early 1970s is that he didn't have more hair in the 1970s, but his hair that he has is now white, and that is the major difference. Okay, so when I started thinking about this talk, I thought, well, if this audience is going to appreciate the science that Joe has done, then I probably need to give sort of an introductory course in biology. And so I started getting slides together for that. And then I realized if I did that, I wouldn't do anything else. So at the risk of maybe losing some of you as I talk about the science, my introductory course is going to be 1 slide long. But I actually enjoy being interrupted. So if I get to something that you don't understand, please, raise your hand, ask me to explain it, or wait till the end, whatever. Okay, so Joe introduced me to the beauty of chromosomes, and this is a human chromosome examined in the scanning electron microscope. It's chromosome right before it's going to divide. It's actually, there's 2 chromatids held together at the centromere, which is a structure that attaches the chromosome properly to the apparatus that pulls the 2 chromosomes apart into the 2 daughter cells. We'll be talking a little bit about the centromere later today. All the chromosomes in higher organisms are linear molecules. That means they have ends. In this case, because it's a replicated chromosome, there's 4 ends, and ends are called telomeres. Telomeres are a subject very dear to my heart, and also the molecular era of telomere biology began in Joe Gall's lab, so I'll be telling you about that. Okay. Now, one of the triumphs of molecular biology was working out the central dogma. Central dogma is already a little less centralized than it used to be. It's evolved over time, but in this very simple rendition of it, this is my introduction to molecular biology. We start with the DNA molecule. You see the double helix, 2 strands wound about each other. As you know, DNA contains within it all the information that tells the cell how to do what it has to do, and that makes you look like your mother or your father, or both, hopefully both. And the information is conveyed in the order of the 4 subunits in the DNA. It's just amazing. All this information that is the basis of life as we know it is just based on 4 different subunits and how they're ordered in a linear array. Now, DNA itself is very precious. It gets inherited every time the cell divides. It gets passed on to our progeny. And so it has to be protected. And so it actually doesn't convey the information directly. It gets copied into RNA, which is also a nucleic acid. So DNA is a nucleic acid, a double-stranded. RNA is single-stranded. And it's a copy of one of the 2 strands for any given region. And the process of making RNA is called transcription. The RNA then gets translated. That means the sequence of those units are read. And each group of 3 subunits codes for an amino acid. And together those amino acids make a chain, which is called a protein. And proteins are sort of the workhorse of the cell. They do most of the work of the cell. So DNA encodes RNA. RNA gets translated into protein. Okay, so Joe Gall was born in 1928. He's exactly 20 years older than I am, which at the time seemed, when I joined his lab, seemed like he was ancient. Now it doesn't seem so old at all. He grew up originally in the suburbs of DC, and then when he was in his early teens, they moved to this beautiful farm, 500-acre farm in rural Virginia. Here's his family. He's the middle of 3 boys. His dad was a lawyer. He actually had to travel a lot, especially once they moved to the country. And you can see this very idyllic environment in which Joe and his brothers grew up in. It was a perfect environment for someone to learn to be a biologist. He actually didn't have to learn. It was kind of just, wandering around, collecting anything in the water, whether it was an insect or an amphibian or plants, etc., and looking at things under a microscope. He credits his mother for being the major force in encouraging his interests. Rather unusual at the time, as he said, he was a pretty, nerd, what would be called nerdy today. Probably wasn't what they called it before, but he was very, just completely involved in learning about living things. And she encouraged this. She got him college-level textbooks so that he could read about it, got him equipment that he needed to continue his studies, etc. Here's Joe, and still at his acting like the naturalist, which he still does today. He collects a lot of the organisms he works on, and he likes nothing better to be out at a pond with his net or out in a field with his butterfly net. And one of the highlights of being a graduate student in his lab, even though we were doing molecular biology and we were working on a molecular level, not with organisms so much, but was to go out and collect. This is at Yale University, and believe it or not, there is rural area around New Haven, Connecticut, and we would go out in the spring into at nighttime and collect frogs. And Joe would sit there and he'd say, "That is," and he'd say what the species name, and he would hear it make it sound. It was really, I have to say, thrilling. Joe is still, as I said, a naturalist. Here he is at a scientific meeting in Costa Rica, absolutely thrilled with this beetle that he found, which was quite a large beetle. At this meeting that we just held in April, Joe's 2 children, he has a son and a daughter, and he has 3 granddaughters, 2 of whom are shown here quite a few years ago. They're now teenagers. And they spoke at the meeting, and one of the things that was interesting to me, because of course I knew Joe as an academic, as a mentor. I didn't know that he in fact instilled so many of the same things, first in his children and then in his grandchildren, about loving nature. They talked about how he was always taking them on walks, and even if it was just, through the city, he'd find some bug somewhere and show it to them. And they all have their bug collections. Here's Joe when he's a young teenager. He talked his parents into getting him a microscope, which, as he pointed out, couldn't have been easy to do at the time because it was, World War II is going on and parts were hard to get. And he actually set up a lab in his bedroom, which you can see here. And the other thing, his older brother built a telescope, which is shown here. And this illustrates for me one of the things that Joe will say about himself. Throughout his career, starting from a very young age, he always used his eyes to see, either through the microscope, to see small things, or up in the skies, just for interest, to see planets and stars, but always observing. Actually, it's a type of science that, observational science, that's not so much in vogue now. It's much more descriptive than what is usually done in molecular biology. And again, he introduced stargazing to me. This was at the party after my thesis defense was held at a friend's house, and he brought his telescope. It was the first time I'd ever seen the rings of Saturn. It was just like magical, so he was always introducing us to these cool things. So when Joe was 17, 1945, he set off to Yale University. He majored in zoology, and at that time, what you did with a degree in zoology is you went to medical school. And it wasn't until near the end of his senior year that a man named Don Poulsen, who was quite a famous Drosophila geneticist, who worked with the Nobel Prize winners who had basically laid out the tenets of genetics, said to him, you can actually study to be a professor at a university. You can get a PhD. And so he decided to do that. He stayed at Yale, and he got his PhD with Don Poulsen. Now, Don Poulsen was a Drosophila geneticist, and Joe wasn't really interested in Drosophila genetics, but that didn't make any difference because Don was the kind of advisor who just let his students do whatever they wanted to do and basically didn't give them much guidance. And I have to say, that's pretty much the path that Joe did when he was an advisor. And people are always surprised about that. They say, well, if he was such a good mentor, how come he didn't give you more advice? Well, his feeling was he gave you opportunity to do science, and that's what he appreciated happening. And he had to find his own research project, which I have to say is not something I had to do when I was in Joe's lab. And after wasting a year trying to find something to do, he stumbled upon something called lamp brush chromosomes, which are being shown here, manipulated by Joe with these are things like very small knives, dissecting needles, and these are chromosomes here. These are chromosomes from the oocyte of amphibians. They're called lamp brush chromosomes. You'll get better pictures of them later because they have these loops that come out from their sides. They're huge. They're actually visible to the eye. And there was just a description of them in an old text, and he decided that he would make that the subject of his PhD thesis to try to understand what was their connection to regular chromosomes. Were they made of DNA? This is really pretty early in the whole understanding of chromosomes. And one of the things he needed in order to study them was a new kind of microscope, a phase microscope, so that he could look at things without fixing them. He wanted to look at them in their natural state without fixing them with chemicals. And he needed to do so with a stage that was inverted relative to where the light was coming from. And he had to make that microscope, which he did with the help of his mom's brother, who was a machinist, and helped him make that. And he took apart that old microscope I showed a picture of and used the parts in making this. So these are some pictures from the paper Joe published on his PhD work. Notice that he is the sole author on this paper, published in 1954. Today, this is almost unheard of, that you see single author papers, especially from a graduate student, because almost all of us put our names on all the papers that come out of our lab. That's the way it's done. But Joe's advisor probably didn't even read his thesis, if I had to guess, and he wasn't going to put his name on it. Here's you'll appreciate how primitive these pictures are. This is a light micrograph, and here's very meticulous drawings of what he thought the chromosomes looked like. When he was only 24 years old, he took a position as an assistant professor at University of Minnesota, and there he is in his office there with his ubiquitous microscope. And when he was there, he did an experiment that first really gave him a lot of attention. This was published in 1963. So remember, the structure of DNA was determined in 1953 by Watson and Crick. So this is very early in the thinking of DNA as the genetic material. And the model that they built was, first of all, a model. It was made out of balls and things like that, and it was based on DNA as a chemical. And the question was that structure found in vivo, and specifically in the complicated chromosomes of higher organisms, such as the lampbrush chromosomes of a newt. And so this is an experiment that he's doing. So the lampbrush chromosome axis is here, and here's one of the loops. And what he's doing in this experiment is he's treating the chromosomes in a very controlled manner with a chemical called DNase. What is DNase? It's an enzyme that cuts DNA. And what he did was to do it and follow the kinetics of it very carefully over time. And what he could then do was determine the rate of cutting was only consistent with the idea that this loop was a double helix that had 2 strands. And the sort of the skeleton of the chromosomes was 2 chromosomes together, and so it had 4 strands. And this was compelling evidence, one of 2 pieces of data that came out at about the same time that made people realize that the DNA model was probably going to be true for higher organisms as well. I show you now just a few other pictures of lampbrush chromosomes so you can just see how incredibly beautiful they are. Here's a more recent picture of Joe's. This is from a 2012 paper using what's called storm microscopy, super resolution microscopy. What you see in green is the DNA, and red is the RNA. Remember, DNA is transcribed into RNA, and these loops are making RNA, and you can see it there, and you can actually see it at the single molecule level when you go up in resolution, or you know how you can see it down there. And here's the most recent paper from Joe, where he's, as I said, he's still extremely active, still doing cutting-edge research, where he's using what's called deep sequencing to understand the information that's in the RNA being made from these lampbrush chromosomes. So absolutely starting from observational work up to the highest tech molecular techniques. Okay, so now in 1963, Joe was enticed to come back to Yale as a full professor. Here you see him with an electron microscope. In the early 60s, electron microscopes were invented. This is, of course, a way of seeing living things, well, not living things, that you can't, because they're in a vacuum, they're dead when you're looking at them, but formerly living things at a much higher magnification. And the government was actually giving these electron microscopes to individual departments so people would start using them. And Joe was one of the early people. He got one at Minnesota, and then actually he had one in our lab at Yale as well. Okay, and then after about 20 or so years as a professor at Yale, he left Yale so that he could spend more time doing his own research. He was becoming very famous, and the demands on his time were very high, and he wanted to go to a research institute. And he did that. He went to the Carnegie Institute, which is affiliated with Johns Hopkins in Baltimore, and that's where his lab is today. Okay, so I've already told you that Joe started working with lampbrush chromosomes, and that he did one of his early experiments was really important in establishing the idea that the chromosomes of higher cells are a double helix of DNA. What I'm going to do now is just talk very briefly about each of 3 accomplishments, which I think are probably his most important contributions to the field of science. So the first of these is the isolation and characterization of ribosomal DNA. So what is ribosomal DNA? First, so ribosomal DNA is DNA that encodes ribosomal RNA. What's ribosomal RNA? Ribosomal RNA is the structural RNA, in this structure called a ribosome, which is where translation takes place. And it turns out about 80–85% of the RNA in the cell is ribosomal RNA. It's a structural RNA. It's not the messenger RNA that has the sequence of information that tells you how to make proteins. it's part of the structure of the ribosome. It's an essential part of the structure of the ribosome where the proteins are made. And because of this, it was the first RNA to be analyzed, because if you're almost 90% of the RNA, it's the easiest RNA to isolate. And of course, like any RNA, it's made from DNA. So it's made from the ribosomal DNA, which is often called the rDNA. Now, the fact that there was so much ribosomal RNA allowed them to have a tool for identifying ribosomal DNA. These experiments I'm going to tell you about were all done before DNA cloning. For those of us who, so DNA cloning started in the sort of late 1970s. And the idea of isolating a gene and determining its, what its structure was like was just impossible because you couldn't isolate them. It's like the proverbial, needle in a haystack. Huge amount of DNA, one gene, you can't do it. But if you have a gene that's copied many times, then you have the possibility of isolating it even before cloning. And that's what Joe did. And what he did was go back to the same stage in development of amphibians, the developing oocyte. The oocyte ultimately becomes the fertilized egg. And these are big cells, first of all. Again, in amphibians, they're, you can see them. They're not like chicken eggs, but they're big. And when you look at them, whoops, sorry, I keep hitting the forward instead of the laser. These are the chromosomes, and then there is this densely staining region, which they had reason to believe was the ribosomal DNA. And the reason is they could see that it was making a huge amount of RNA. And because the oocyte is so big, it has to make all this protein. I mean, because it's huge, it needs a huge amount of ribosomes. And there's not enough ribosomal DNA to make enough ribosomal RNA to make the ribosomes. So what happens in amphibians is they amplify. That is, they make extra copies of the ribosomal DNA. Normally, every time a chromosome copies its DNA, you start with one copy, you end up with two. This is a case where at the end of the process of amplification, you have 1,000 times more rDNA per cell than you do in a normal cell. And this, first of all, it was the realization that this differentially staining material, this is stained with something called fulgin, and these black dots are indicating that it's making a lot of RNA, its radioactivity, was part of the realization that this was a special kind of DNA and rDNA. And then he realized that he could isolate the ribosomal DNA on something called a cesium chloride gradient. Now, what's a cesium chloride gradient? God, I did a million of these when I was a graduate student. These are something that you run in an ultracentrifuge, and it allows you, which, as its name implies, is a very high-speed centrifuge. And it can separate DNA of different subunit complexity. Remember, there's 4 subunits in DNA, A, G, C, and T. And if you have a lot of G, you tend to be lower in the gradient. These are the numbers from the gradient than things that have less G. And it turns out the ribosomal DNA is very G-rich. And so you can get it as a separate peak compared to the rest of the DNA. So in orange is before amplification. Most of the DNA is not rDNA, shown here at this G-rich peak. But with amplification, most of the DNA in those cells is actually ribosomal DNA. So he could isolate it by separating it on these gradients, and he had pure rDNA. He had, in fact, isolated a gene, the first gene to be isolated. So that's pretty cool. Okay, so that's the first thing. Everybody get that? Is that sort of? Okay. What's very, I think, interesting, when I was thinking about all these, is that you can just see the continuity in his career and how different experiments flow from one to the other, which to me is very appealing. Joe always said, he told his students, never spend the rest of your life redoing your PhD thesis. And he said, and that's exactly what he did, and he's still doing it, working on these last first chromosomes, trying to figure out what they are. Okay, so the next one is the development of a technique, which is still used today. It was developed in his lab in the very late 60s, early 1970s, called in situ hybridization. And this is a method that allows you to say, if you're looking at this big piece of DNA or this, part of a chromosome, where is gene X located on that chromosome? Because a lot of times you want to know that. So he tried to do this for about 10 years before he finally got it to work. And the breakthrough was realizing, because he kept getting negative results, he couldn't get it to work, but he didn't know if it was failing or that he just wasn't sensitive enough. Maybe there wasn't enough copies of the DNA for him to see. So he said, let me use these egg nuclei that have 1,000 times more rDNA. And if my technique is working, and what he's doing is he has labeled ribosomal RNA, radioactively labeled ribosomal RNA, and he's trying to get it to bind back to the DNA from which it's transcribed. And he knows where that DNA is. It's this cap here. And so he's looking at oocytes at different stages in development. And he's hybridizing them for ribosomal RNA. And the real key here is this huge hybridization in the cap, which he already knows is the ribosomal DNA. And it's hybridizing to ribosomal RNA. So his conditions are correct. He can get the RNA to hybridize to the DNA. Now, this was cool technically, but we already knew the answer, so it wasn't fundamentally interesting. What you'd want to do is to find out where is a certain DNA located on the chromosome that you don't know anything about its location. The problem is this is still before cloning. So you had to have a DNA that you could physically isolate. And I neglected to say, this work was done with Joe and his graduate student, Mary Lou Pardue. So Joe and Mary Lou next applied this technique to mouse chromosomes. And in this case, they're isolating about 10% of the DNA in every mouse cell is called satellite DNA. Why is it called satellite DNA? It's because it forms a peak on a cesium chloride gradient away from the rest of the DNA. Nobody knew anything else about it other than that fact. So they labeled that, they isolated that DNA, they labeled it, and they hybridized it back to, these are just regular mouse chromosomes, and it hybridized at a very specific place. And that place is the centromere. It's a little hard to see it here compared to that first picture I told you, because mouse chromosomes are what are called acrocentric. They have one long arm, and the second arm is really, really short, so you don't see the centromere between the two arms. But this is one chromatid, here's the other, and here's the satellite DNA. And every one of the chromosomes has that sequence. Here's a more recent rendition of this technique, now called FISH. Instead of using radioactivity, which has the problem that, first of all, it's radioactivity, and you have to wait for the exposure. But in addition, when the radioactive atom decays, it moves a certain distance, so it decreases your resolution. So if you didn't, here you're just using a fluorescent probe that when you shine UV on it, it lights up. And it's attached to, in this case, your DNA sequence. And what we're looking at here is human chromosomes that are subjected to in situ hybridization with fluorescent probes, or FISH, looking for both the centromere, which is here in red, and now you can see the short arm and the long arm of each chromosome, and then the telomeres, which are there in blue. So two different sequences hybridized. And this method is now sensitive enough that it can see single copy genes. It's really important in terms of studying genotypes of, for example, tumor cells, when chromosomes have changed in all kinds of ways, and you want to see if gene X has now moved near gene Y, and you can see it with this method. Okay, so that was the second big contribution I wanted to highlight. The third is the first molecular characterization of telomere DNA. Now remember, telomeres are the ends of chromosomes. And for a long time, people just thought, it's like a piece of string. The end is just where it stops. But actually, the end is actually a structure. And people use the comparison with shoelaces. You know the plastic tip on the end of the shoelace? Anybody know what that's called? It's a good crossword puzzle word. An aglet. And it was actually Newsweek magazine that first did the comparison. They said the telomere is like the aglet because if you lose that plastic cap, what happens? Your end of your shoelace unravels, right? And you start losing information. The telomere is a structure. It's like the aglet. And if you lose the telomere, it's like the worst thing that can happen to your chromosomes because it is rampant genome instability. It's considered one of the initiating events in virtually all tumors, is loss of telomeric DNA. So this is an important question, although the connection to human health and disease wasn't realized until the telomeric DNA was isolated and characterized. So this is a picture of Joe at the time he started this work. Now, this is a picture over here of one of Joe's favorite organisms, and it truly is a beautiful single-celled organism. It's a ciliated protozoan called Tetrahymena. And so these little things coming off are cilia, they beat, and it's how it moves through. It's a motile organism. And Joe first studied these from pond water from his house, not this species, but a related species. He actually wrote a paper on it while he was still in high school. But ciliates and ciliates are big cells too. There is kind of a theme in this. A lot of the first work on chromosomes and DNA starts with structures that are big because you can get your handle on them. They often have a lot of DNA. So Tetrahymena is almost visible to the eye. Paramecium, maybe you remember paramecium from using it in school. That you often can see with your eye. This is a slightly smaller relative. And all ciliates have a very unusual, this is a single cell. This whole thing is a single cell. But it has two kinds of nuclei. It has a macronucleus and a micronucleus. The macronucleus is big. The micronucleus is small. What ciliates do is actually combine in one cell sort of what we do in a whole body. The micronucleus is the germline. It's the equivalent of eggs or sperm. It does not make any RNA. It only participates in the exchange of information after cells mate. So it's kept small and protected and has just normal chromosomes. The macronucleus is huge. And instead of having 2 copies of every chromosome, it has hundreds of copies. And it's made in a rather haphazard way, but it doesn't make any difference because it's not the genetic nucleus. It's not going to pass it on to the next generation. And all the transcription takes place in the macronucleus. So every time Tetrahymena mates, which it doesn't do very often, but when it mates, then you get rid of these nuclei. You get a new micronucleus that's a fusion from the 2 cells that are mating, and that generates a new macronucleus. And the macronucleus is generated by making lots of copies of the micronucleus and chopping it up into little pieces. It looks completely haphazard, but it's not. So you get lots of pieces and you get lots of ends. That lots of ends is important. If you're a human cell and you have 23 pairs of chromosomes, so you have 46 chromosomes, you have, let's see, what's times 2. 92 telomeres per cell. But some of these ciliates have literally 10 million telomeres per cell because they chop up their DNA into such small pieces. Now, tetrahymena is not quite as wild as some of those other ciliates, but it still has lots of ends per cell. Okay, now, he read a paper, an old paper. Joe is also an amazing historian of science. And he's always going back in the literature and finding things that were already known and then working on them at the molecular level. So there was a description of the nucleus, of the macronucleus, that made him think that it might have amplified ribosomal DNA, or DNA, just as you see in amphibians. And so he got these organisms, he isolated the DNA, he ran it on a cesium chloride gradient, he got the rDNA, and he then looked at it in the electron microscope. So this is an electron micrograph of isolated ribosomal DNA from the ciliate, and there were linear molecules and there were circles in there. And there was clearly a lot more ribosomal DNA than anything else in the cell, any other kind of DNA species. Now the first thing, so he did that work by himself and published it just by himself. This work he did with Kathy Carr, a graduate student in the lab. He wanted to determine the structure of the rDNA. And what he found out, these are now looking at the linear molecules. This is a linear molecule looked at in the electron microscope. If you're wondering what these small circles are, these are markers. They're Phi-X174. It's a bacteriophage that you just use as a size marker because you know exactly how big it is. Okay? And he's seeing that there's this funny structure in the middle of the molecule. So with a bunch of experiments like that, he and Kathy were able to figure out that these 20 kb, 20,000 base pairs, so 20,000 units of those 4 units in a row, is actually a huge palindrome. What's a palindrome? It's something that reads the same from one end as from the other. And I've just put on letters here, so if you're going from the left end of the molecule, A, B, C, D, or if you go from the right end, A, B, C, D, right? And the result of that is that this half of the molecule is actually complementary to that half. So if you separate the 2 strands and let them come back, they'll form a half-length molecule. And it's got a center of symmetry. So that was pretty interesting. And actually, when there were 2 people working on that experiment at the time, Kathy and Susan Hodson, who goes by the name Suze, who's actually a pediatrician now in the Princeton area. She was a technician in the lab at the time. So they figured out the structure when Joe was in Europe. And this is before email or anything like that, of course. And so Rob Granger was a postdoc in the lab, and he was from Berkeley. And we all know what Berkeley was like, you know, in the late 60s, early 70s. And he said, we have to send him the result in a palindrome. So with his Berkeley influence, he got sex-aware error waxes. And then this is also close to a palindrome. GL is the name of the species. And her name is Kathy Carr, so she's KK, she's the same. And they spelled her name a little differently, but made her a palindrome too. So that's the note. And he said it took him a couple hours to figure out what they were talking about. But they didn't hear back from him. They thought, you know, this is really exciting and completely unexpected. Well, the reason they didn't hear right back from him, it took them 2 days to figure out a palindromic answer. And this is actually a copy of what the telegram that he sent. And so, you know, he even palindromed his name instead of JGG, JG, and you could see he turned it into his answer into a palindrome. So that was pretty interesting. The next part of this story, though, to get to the end of the chromosomes, we're just on our way to the ends, is when Liz Blackburn joined the lab. Liz Blackburn is from Tasmania, Australia. And then she did her PhD in chemistry with Sanger at Cambridge University in England. Sanger would eventually get his second Nobel Prize for determining how to do sequencing of DNA, called Sanger sequencing. There's Maxim Gilbert DNA sequencing. Sanger, of course, did Sanger sequencing. And Liz did her PhD with him, and she knew how to sequence DNA. She was maybe one of 5 people in the world who knew how to sequence DNA. She had a PhD in chemistry, and she knew no biology. So she came to Joe's lab, and she said, I want to sequence something important. And he said, oh, well, you know, because the problem was they were just sequencing these little viruses, these bacteria viruses, because you couldn't clone anything. This is before cloning. So he said, maybe you should sequence the ends of ribosomal DNA. No one knows what the end of any molecule looks like. And you could isolate them by the methods I told you about. You know, there's a lot of them in the nucleus. You put them over those cesium chloride gradients, and then you sequence them. And this is what the sequence was. There was about 400 base pairs. It was actually, you know, about 300 base pairs on each end. The ends were mirror images. The strand running 5' to 3' from each end was C, 4 C's followed by 2 A's. And then there were about 50 copies of that. That was kind of boring because it couldn't encode anything. It's just C4A2, C4A2, C4A2. But it was the first telomere DNA to be sequenced. So, and the only reason you could sequence it, because Joe knew how to get the ends, the natural ends, without going through cloning. The second telomere DNA to be sequenced was sequenced by a guy named David Prescott, also working with a ciliated proteome. Again, he could directly sequence the ends of the DNA because of this weird genome organization. And the sequence wasn't C4A2. It was C4A4. Pretty similar, same orientation. And then more DNAs got sequenced, and it was found that all you really needed to be a telomere was to have G-rich DNA in the strand running 3' to 5' towards the center. It didn't have to have no A's in the strand, for example. It had to have the G tail. And in fact, it's now known that this G strand is extended to form a single-stranded G tail. And it's that G tail that is really critical for the structure of the telomere. So telomeres do a lot more work from a lot of different people. It became clear that the regulation of telomere DNA is a key part of human biology. No, I just realized I'm not paying any attention. Oh, I'm going to run out of time here. Really, I got to go faster. The Nobel Prize was given in 2019 to Liz Blackburn. Remember, she was the one who sequenced the telomeres, her graduate student, and a guy named Jack Shostak, who had done one experiment collaborating with Liz. A lot of people thought Joe should have gotten it instead, but he did get to go to the Nobel Prize ceremony as Liz's guest. Okay, you know, I've really screwed up here. So now I was going to try to talk a little bit about what it's, the aspect of Joe that has to do with his encouraging women. And I think what I just want to say, as I'm going to go through this a little quickly now, so I was an undergraduate at Cornell, then I went to Yale University, and then to Princeton. At Cornell, I had one female teacher in 4 years. There was no female faculty member in my department where I did my research. At Yale, there was one female in the department. She could get tenure. I came and did a postdoc at Princeton for a year. There was one female in the department. She didn't get tenure. Actually, I thought I had one more down here. I should have one more. And I was a postdoc for 2 years at University of Washington in genetics. There was one woman in that department, and she got tenure. Okay, but it was not it was not a hospitable place for women in science. There rampant sexism, rampant harassment. I don't think any one place there's the other one, was better or worse than any others. Okay, so this is what Joe looked like when I started in his lab. And these three people, myself, Sharon Endo, and Pat Tequila, were all graduate students that entered the Yale Biology Department in 1970. We did a year at Yale while Joe was on sabbatical and hated everybody we rotated with. So when Joe came back from sabbatical, we were all on his doorstep, and he accepted all of us into the lab, and we all got our PhDs with him. And one of the things I think that is really interesting is that all of us are professors at major research institutions. This is not a coincidence. I think it has a lot to do with the fact that we got our PhDs in Joe's lab. So, I mean, it was a weird time in science. I mean, you'd go to a scientific meeting. First of all, they kept calling us Gall's girls. And we weren't the first females in Joe's lab. There had been a tradition of having females in the lab, as you'll see in a second. But one of the stories I loved the best, we were all at a meeting together in Tennessee, and Sharon was asked by a very famous scientist, this kind of just incredibly lecherous voice, so what does Joe have that all you girls want to be in his lab? So Sharon said, well, you know, when you go into Joe's lab, office, and you sit down and talk with him, and he goes to shut the door, and he goes and shuts the door so that he can hear about your research. We talk about our research. I thought that was a pretty good answer. So why did the three of us succeed? In part, we were really lucky because we had role models. Shown on the left is Joan Stites. Joe took this picture of her during a partial eclipse of the sun in Minnesota, which is what those things are. Joan was an undergraduate in his lab. She was planning, brilliant person, sort of Nobel Prize quality person. Her husband did get a Nobel Prize 2 years ago, Tom Stites. But in any case, Joan was just assuming she'd go to medical school because she knew there were some female doctors, but she knew no female scientists. But before going to medical school, she spent a year working in Joe's, not a year, she spent a summer working in Joe's lab, and he convinced her to get a PhD. So she goes to Harvard, and she goes to Jim Watson's lab and says, I want to work in your lab. And he said, I'm sorry, I don't take female students. And she said, this is, I don't actually know if this part of it's a true story. I do know up until then it's a true story, but this part may or not be apocryphal. And she said, let me work in your lab for 1 month or 2 months, and if I'm not the best student you've had, kick me out. And so she got her PhD with Jim, and after that he always took female students. She went from there to Cambridge and then came back to Yale as an assistant professor in 1970, the second female in the department. So she was an undergraduate with Joe at Minnesota. Susan Jerby was a graduate student. I didn't overlap with any of these people, although she came to Yale at the same time I did, although as a faculty member. Susan, who's now a good friend, was a graduate student, and she became an assistant professor at Brown, where she still is, in 1972. And Mary Lou Pardue, whose work you heard on in situ hybridization, became a faculty member at MIT in 1972. Both of them were the second women in their departments. And the reason they got jobs was because at about this time, HEW started investigating universities and saying, you are discriminating against women, and you are going to lose federal research money if you don't hire women. So they all hired a woman. And it took until many years later before those numbers really improved. But I did have role models because these people would come back, Susan and Mary Lou, and talk to us, and you could see, you know, people went on and they got jobs. This is just to show you what some of these people look like in 1983. There's Susan and Mary Lou. And there were other talented and ambitious people in the lab when I was there. Many of them were women. Mary Lake Poland was a postdoc. She went on to go to medical school. She was head of obstetrics and gynecology at Stanford for many years. She's now at Columbia. Amy Bakken, faculty member, University of Washington, now retired. Gaylor Hartford at Albany. Kathy Carr, you saw her work earlier at Marquette. Liz Blackburn, postdoc She was our Nobel Prize winner now at UCSF. Jeannie Bennett was an undergraduate who collected embryos for me for my research project. She's now an amazing physician, MD, PhD, at University of Pennsylvania Children's Hospital. She and her husband together conducted one of the most successful gene transplant experiments to correct for a kind of hereditary blindness. Very, very talented person. I just actually saw her. I'd given a seminar there, and she came up to talk to me. I actually would never have imagined she liked to lie out in the sun and get a tan. But boy, she's a smart and talented person. This is just to point out to you that times have changed. In my department here at Princeton, and I've been here now for 18 years, and this has not always been true of my department, but it's very impressive improvement, because I have to tell you, there's not a pipeline problem in the basic sciences. There's been close to parity of women getting PhDs in the life sciences for about 40 years. Pipeline is not the problem. But you can see that my department has 41% tenure track faculty female. Most of them are married. It used to be if you were lucky enough to get a job, you didn't get married. You certainly didn't have children. Our faculty, female faculty, have children at about the same frequency as our male faculty. And there's this idea you have to have your children after tenure or you never get tenure. It's certainly not true at Princeton. I have 2 children myself. As you know, Shirley Tillman, our former president, who's a member of our department, has 2 children. So here's another reason that Joe gave you independence and he gave you credit. These are examples of all people who published their work while I was there under just their name. This is extremely unusual. Joe said, I didn't do the work, you did the work, you did the thinking, you get the credit. That was also faith in you. He also cared about the science. He actually said, he believed women and men have an equal chance of succeeding, equal chance of failing too. He thought most people failed, but women were not more likely to fail. He also once said, people thought I only had women in my lab because, the women were the famous ones in it. He said, I have roughly equal number of men and women, but the women were much more successful. Yes. Here you see Joe reading Mendel's copy of Darwin. Here's some of the awards Joe has won. I especially like these awards that have to do with mentoring. You can see him here receiving some of these awards. This is an honorary degree from one of the oldest universities in the world in Prague. This is a Lasker Award. This is often thought of as the forerunner to the Nobel Prize. He did not get the Nobel Prize that came after this, but he did get the Lasker. Here you see, for example, Joan Stites, still at Gall, Mary Lou, still at MIT, Susan, still at Brown, with Joe at the Lasker ceremony. His children getting the Women in Cell Biology Award, Senior Woman Award, for all of his work. I've won that as well, but Joe's the only male to have received it. There he is getting the Horowitz Prize, and I'll end by just saying how happy and grateful I am that I got my training in the lab of such a great scientist, but also someone that allowed me to reach my potential. And I'm happy to take questions. I'm sorry if that went a bit long. Yes?
Commented on the role of telomeres in cell cancer, but the work that I was following, a lot of this stuff is telomeres. Can you comment on that? Is that still very much?
Absolutely. So, in fact, the enzyme that makes telomeric DNA is called telomerase. And it's present in incredibly low amounts in almost all organisms, but certainly in humans. It's not expressed at all in most cells in our body except our stem cells and cancer cells. And stem cells, what is the cause of aging in many situations is running out of stem cells. And stem cells run out of telomere DNA. Because even though they express telomerase, it doesn't keep up with the rate of cell division. So I would say the link between aging and telomeres is stronger than ever. Very, I would say even stronger than there's a strong correlation between being a tumor cell and being turning on the activity that makes telomeres, but how causative that is not always so clear. Aging, I think, is much clearer. Yes?
When you study the DNA of protozoa, and you compare it to human DNA and evolution, do you see any similarity?
Oh, you see a huge amount of similarity. There's certain genes like cytochrome C or actin or histones. I mean, it is very hard to not believe in evolution when you look at DNA sequences. Now, on the other hand, ciliates are off on a tree all by themselves. They have a different, they don't even have quite the same genetic code as bacteria do and eukaryotes do, higher cells do. So they are unusual, but there are certain genes that are absolutely highly conserved. But the organization of the DNA is very different. Yes?
What accounts for the large size of the chromosomes that Professor Gall was looking at?
Good question. So the answer to that is still not really known. One of the questions on my general exam in 1971 was to explain the C-value paradox. C-value paradox is the fact that organisms can have such a different amount of DNA per cell in a given organism. One organism might have, that's closely related, X and Y might have 10 times different amount of DNA per cell, and yet they're closely related. So it turns out amphibians just have a huge amount of DNA per cell, and so their chromosomes are bigger to start with, but they're extended in this particular stage of development in the oocyte so that a very large fraction of the DNA can be transcribed. And the reason Joe still works on lamp brush chromosomes is that it's not really understood the significance of that.